Chapter 14, Lipid Metabolism, Commentary C. In this portion of the commentary, we're going to go ahead and talk about fatty acid synthesis. So this is assuming that we have plenty of fatty acids, such as from that cheeseburger. Yes, the body still has to break it down into individual glycerol monosaccharides. But if you have plenty of it, well, if you have any of it, automatically you can assume that it's going to go ahead and form triglycerides. So in synthesizing our fatty acids, you need the proper enzymes, but it's not simply the reverse of the fatty acid enzymes using metabolism. They use different enzymes. Although they essentially do link together acetyl groups from acetyl coenzyme A to form the desired fatty acid. The fatty acid synthesis takes place in the cytoplasm as opposed to the breakdown of the fatty acids which occur in the mitochondria. So notice the big difference here. One occurs in the cytoplasm, whereas the other one occurs in the mitochondria. There is a high energy requirement for synthesis of fatty acids, but it's assumed by the body that if you have plenty of fatty acids that are available, then automatically you have a high energy content to begin with, and don't mind wasting a whole bunch of ATP to synthesize the fatty acids. Although the acetyl acid is made in mitochondria, it needs to be transported to the cytoplasm. So the first thing we need to do is get the acetyl-CoAs into the cytoplasm. So what happens is the acetyl-CoA binds with available oxaloacetate and it forms citrate. The citrate is then transported to the citrate transport system. We don't have enough time to go through that, but it's transported to the cytoplasm in this form. So make sure you know that. The acetyl-CoA and oxaloacetate are then regenerated back into their original forms when the citrate is broken down. Now that we have the acetyl-CoA in the cytoplasm, the acetyl-CoA is going to form the desired fatty acid. In this case, palmitic acid, aka palmitate. So palmitic acid, palmitate, same thing. Or other types of fatty acids, desired fatty acids. Other non-essential fatty acids. Now, I'm not going to go through the big process of how this occurs. Just know that it's a multi-enzyme complex made up of 6 enzymes all clumped together along with an additional protein called a acyl carrier protein. This is the general before and after. Yes, there are multiple steps, but that's the overall reaction that you're responsible for knowing, since we are short on time. Fatty acids are then converted into triglycerides and they're stored as adipose tissue. Please keep in mind our bodies do not have the enzyme to convert acetyl-CoA directly to pyruvate, so we can't sit there and turn fat to sugar. We don't have those specific enzymes. We can't turn acetyl-CoA into sugar. So, amino acids metabolism. Now we're going to get into some common pathways for amino acids. First off, here is a general diagram. Here we see proteins that are digested. They immediately form amino acids. Now, there's a constant turnover where the amino acids are synthesizing into important body proteins. This anabolic process we've already talked about. At the same time, proteins that have basically lived for a while, they get older, they start to break down, so the body digests them and recycles them back into amino acids. So the constant breakdown and synthesis of proteins to amino acids, we refer to this as the protein turnover. So when we're breaking them down, we need water to do this, which is where we get the term proteins are hydrolyzed. The protein turnover rate is the rate that this occurs, and so it's the time it takes for 1/2 of the protein to be turned over. So that means to go from amino acids to proteins back to amino acids. So you can go through the rest of this. These are just the highlights that you absolutely need to know. Um, let's go ahead and assume that we're at this point right here, and now we're going to go ahead and explore amino acid catabolism. Notice in catabolism, we're going to go ahead and break down our amino acid into a carbon skeleton and remove that amine group into a form called ammonium ions. The carbon skeleton can then be inserted into the citric acid cycle via multiple pathways, which we'll talk about in a second, and the ammonium can be converted into urea for excretion. Now, in order to catabolize it and create these leftover carbon skeletons in ammonia, we have three stages. The first one is transamination, the second one deamination, and the third is urea formation. I'm going to go ahead and show you the overall reaction for the transamination from an amino acid to glutamate. Although we also have amino acid transaminations to aspartate, these are not shown in these particular slides. Why? Because we are showing you enough already, so I figured I wouldn't show this to you. So I'm just going to go ahead and focus on glutamate in this instance. Glutamate is then going to go ahead and lose its amine, forming an ammonium and carbon skeleton, at which point it's degraded and inserted into the citric acid cycle, which we'll get into in a bit. The ammonium is then converted into urea. So again, we're not going to be focusing on aspartate in my slide summaries, nor will it be focused on the final. Transamination, we are going to focus on the glutamate. So here's a nice example along with enzymes. Know all of this. Here we have a amino acid. Here is the amine. Notice it's in water, so it's protonated. So it's actually an ammonium ion. And what's going to happen is we're going to transfer that ammonium over to the carbon containing the carbonyl group. And that carbonyl group is going to be transferred back over to the donor amino acid. So because it's transferring, we need an enzyme that does this called a transaminase. So thereby leaving a new keto acid and a new glutamate. Now in deamination, we're going to go ahead and remove the ammonium ion. So let's go ahead and highlight this here. In doing so, we're going to go ahead and regenerate an NAD to NADH. Let's go ahead and go through this really quick. Using NAD along with an available water molecule, the ammonium is going to come off. Keep in mind it's already protonated from the water, and it's going to be, when it's by itself, it's going to be protonated again, forming a brand new free-floating ammonium ion, NH4+. At the same time, you're going to have an incoming water molecule, and this is where it gets a little bit complicated, but the oxygen from the water molecule is going to go ahead and form a brand new carbonyl group with a vacating hydrogen. As far as this class goes, you can just go ahead and assume that the hydrogen that's going to be vacating is going to be attached to the NAD, forming NADH. And then you have a leftover hydrogen from the other hydrogen from the water molecule. It finally leaves a alpha ketoglutarate, and make sure you understand that in this process it's going to be catalyzed by the enzyme glutamate dehydrogenase. Cool stuff. Again, we're not going to be focusing on what happens to aspartate just due to time limitations. Now, in this form, ammonium just can't sit there and leave the body. It needs to have a transport vehicle, so to speak. So what happens is it's going to react with 3-ATP, water, and aspartate. In the process, the ammonium is going to go ahead and attach as a to the aspartate, and it's a little bit complicated, but what's going to happen is when it attaches to the aspartate, it's going to form it's going to form a diamine. Technically, it's a diamide if we want to be specific about it. So you're going to have two amines attached to a carbonyl group, so technically it's a diamide. Bottom line is you have two amines on there, so it's easier for it to get rid of two at a time. In this form, it's called a urea. The leftover portions of the aspartate is then forms what's called a fumarate, which can then be recycled to another pathway, such as the Krebs cycle. Keep in mind it actually does take energy for this to occur. In summary, the ammonium is going to be transferred to an aspartate, which breaks down, forming a urea. We call this process the urea cycle. It's actually really complicated with multiple steps, so this is the before and after general reaction. This occurs in the liver, but then diffuses to the blood and then is filtered by the kidneys, at which point it is excreted. Notice how we've been focusing on the formation of urea. Now we need to focus on the leftover carbon skeleton. The leftover carbon skeletons are degraded to pyruvate, acetyl-CoA, or acetyl-acetyl-CoA. There are other citric acid cycle intermediates, but these are the first big three. Know all of them. Now what we like to do is we like to go ahead and break them down into groups. If ultimately they like to form acetyl or acetoacetyl-CoA, then we call them ketogenic. However, if they form an intermediate which goes directly into glucosynthesis, such as pyruvate, or a intermediate which goes directly into the citric acid cycle, we call this glucogenic. So think ketogenic, anything related to acetyl groups, such as with fats. Glucogenic, anything related to glucose, carbs, as well as the citric acid cycle. These definitions can get kind of complicated, so I've summarized them here. K for ketogenic. See how they go directly into acetyl-CoA or acetoacetyl-CoA. G for glucogenic. Here they go directly either into pyruvate or they perform an intermediate in the citric acid cycle. You're responsible for giving examples into the differences between ketogenic and glucogenic amino acids. Just as we have essential and non-essential fatty acids, we also have essential and non-essential amino acids. Non-essential being ones we can naturally synthesize. Essential we cannot, so we need to get from our diet. So you are responsible for putting these on your cheat sheet, aka memorizing them. This part over here shows you the breakdown. It shows you the synthesis of various starting glycolysis, citric acid, or amino acid intermediates in order to create our non-essentials. So, but I'm not going to test you on this part. I am going to test you on this here and test you on this. And this